COVID-19 map walkthrough. We appreciate you taking the time to learn about the COVID-19 competitive landscape via our map tour today. And we really hope that the map tour will really kind of help you navigate the pandemic response in your respective positions. So I just wanted to start with a few housekeeping items. So as you'll see on your Zoom screen, there is a question and answer button below. And we really encourage you to ask questions sort of in real time throughout the presentation. And we will try to address those questions, both verbally, and then also we can type out answers to you. So please utilize that. There's also an upvote button. So that will help us prioritize questions in the event that we get a lot. So please feel free to upvote any questions that are particularly interesting to you. We'll also leave the presentation open for about 15 minutes after we conclude so that you can peruse the question and answer section if you're interested. Also, just would like to note that this presentation to be taken is off the record, and if you would like any quotes pertaining to the presentation, please do reach out and we can supply them. And lastly, we would be really excited to engage with you further after this map tour. So if you are interested, please do reach out to me and we can get a follow-up meeting scheduled to talk about sort of the response that they're looking at. We'll also be scheduling additional map tours in the future. We'll have that information on our website. Obviously this is a very rapidly evolving field. And so we try to keep up to date with that, and we'd like to share, those new findings as we will today in those subsequent map tours. So with that, again, thank you so much for joining us, and I will hand it off to Peter Kolchinsky, the founder and managing director at RA, and he will kick things off. So thank you.
Thanks, Alyssa. So welcome everybody. This is now our third map tour, and the field continues to evolve. There was just news that came out from Novavax, and we scrambled to integrate it with the data from other companies. So as we get through the vaccine portion of the tour, we'll pause there on the data to look at the comparative effectiveness, tolerability of all of the sort of first wave of vaccines. Just to put into context what you're gonna hear, we like to put the bottom line at the top. And the takeaway from everything that we're seeing on the vaccine and the drug side is we still don't have great treatments. It's really important to try to prevent infections. So social distancing till we have a vaccine is going to be critical. It really puts into question all the plans people have for reopening schools. The way things are going with sports teams, it really doesn't look very promising for schools. So we got to get vaccines out there as quickly as possible. And there's a number of companies that have vaccines in clinical development that have generated neutralizing antibodies. We're going to be talking a lot about neutralizing antibodies. Those are the functional good antibodies that you need that stop the virus from infecting cells. And to really end the pandemic, you need to get a high enough neutralizing titer for some period of time, maybe 6 months or a year, so that we can really knock back the virus. It doesn't mean we'll make all COVID infections go away. And there will no doubt, I think after the initial pandemic response, there'll be a period where we're going to be, well, forever after, we're going to be giving booster shots of COVID vaccines. And so the question for getting out of the pandemic is, in the near term, what are good enough COVID-only vaccines? And right now we have a bunch of vaccines that generate some amounts of neutralizing antibody titers. And we will find out from much larger clinical studies that are just starting up how protective those vaccines are likely to be. So if you enroll 30,000 patients, half of which get a vaccine and half of which get a placebo, then if, let's say, 1% of the people in the placebo arm get an infection, that would be 150 patients out of 15,000. And the question will be, how many fewer patients than that, subjects than that, got infected in the vaccine arm? If it's 75 versus 150, then you would say the vaccine is 50% protective. If it's 50 versus 150, you'd say it's 2/3 protective. So we need those data to find out just how effective these vaccines are. And I think we're going to be getting them, several months from now. And those will be the basis for ultimately approving the first wave of vaccines. And then you're going to see on our charts how we look at who's making how many doses to try and envision when might we have enough doses to get the US and Europe and the rest of the world out from under this pandemic. And then later on, when we're in the period where we're going to be getting booster shots, maybe once a year, it actually makes sense to combine those booster shots with flu vaccines. So we're going to be looking at what are the most tolerable vaccines, because what you're not going to want to take are vaccines that hurt a lot. And the thing you'll see from a lot of these, initial COVID-only vaccines is they're really uncomfortable, and arguably more uncomfortable than the most uncomfortable commercially available vaccine that's out there. So it's going to be important for figuring out who's going to serve the world's need for a regular booster shot of COVID, and particularly one that can be combined with flu vaccines, because you're not going to want to get 2 shots if you can avoid it. And by the way, in the future, we're going to be thinking about flu vaccines as a way of minimizing COVID-like symptoms, because when you experience a fever or a cough, you're not going to think flu anymore. You're going to think COVID. And so making sure everybody gets flu vaccines in order to reduce the chance that you're going to see students or workers experiencing COVID-like symptoms and freaking people out is going to be pretty important. So we'll talk about which are the vaccine platforms that are most likely to yield a combination flu plus COVID vaccine. It may be that vaccines are not going to be perfectly effective in the long run at completely eliminating people getting COVID. After all, there are older people whose immune systems don't respond really strongly to a vaccine, and we're going to need treatments. Even now with flu vaccines, granted only about half of America gets them, you have anywhere from 20 to 60,000 people a year in the US who die from flu, right? So that's the kind of problem that merits drugs. So we're going to talk about drugs that are in development to try and treat infections. We've got only 2 with emergency use authorization. We'll talk about some of the others that are in development. And then we'll also talk about the screening and testing paradigm that's going to help us identify people who might be at risk, who in fact are infected, and who have recovered from infection, and whether or not they're still immune. So we'll cover all 3 parts of that. So I'll pass it on to my colleague Shanshan, who will take you on a tour of the vaccine side of the map.
Great, thanks for the introduction, Peter, and really summarizing the key learnings from the COVID-19 map that we've put together. So my name is Shanshan, and I'm a Tech Atlas Associate at RA. And today I'm going to walk everyone through the left side of our COVID-19 map, which covers the vaccine development landscape. And as you can see from a first glance, this map is really large and can feel overwhelming. So the goal of today's walkthrough is just to help everyone familiarize themselves with the structure of our map and how it's been designed to address key questions around both vaccine development on the left and therapeutics on the right, which one of my colleagues will take you through later today. So for vaccines on the left, the 3 key questions our map really addresses is, firstly, out of the almost 200 vaccine candidates out there, which technologies and platforms we think are most likely to work? Secondly, out of the vaccines already in clinical testing, when might we expect these vaccines to come online, and how much will be available? And then lastly, based off of recent and also upcoming data releases, how do these vaccine candidates stack up against one another? And then what might good data look like? And so I'm going to take us through the map to address the first 2 questions and then hand it back off to Peter to really look at some of the data that recently came out in detail. So starting off, before we can assess any COVID-19 vaccine, we have to understand what a successful vaccine looks like. And on our map, we have a number of infographics, such as this one, that teach us how SARS-CoV-2, which is the virus that causes COVID-19, enters the host cell through interactions between the spike protein on the viral surface and the ACE2 receptor located on the host cell membrane. And we know from various vaccine development efforts that neutralizing antibodies against the spike protein that interfere with this interaction are protective. And so protect us from infection and developing severe disease. And this is why most of the vaccine development efforts out there are going after this specific antigen. However, it's reasonable to expect, even after developing neutralizing antibodies, that protection or immunity against COVID-19 will wane over time. This is supported by our previous experience with coronaviruses that cause the common cold, which we never really developed permanent immunity against. And also, when we look at the sera from patients who have recovered from COVID-19, the levels of neutralizing antibodies, or antibodies in general, decrease and level off over time, which means, like Peter reiterated at the beginning, that a successful COVID-19 vaccine comes in two flavors. First, it must help us get out of this pandemic setting. So back to the new normal and provide sufficient protection. And then second, it must be amenable to redosing or or maybe even seasonal administration. So now that we know what to look for in a successful COVID-19 vaccine candidate, we can actually look to the vaccines in development. And on the map, we've separated these out into two main buckets. All of the vaccines on the top half of the map are based off of proven and widely used platforms. So this includes protein subunit approaches and whole virus approaches. And then all of the vaccines on the bottom half of the map, which we'll get to later, are the novel platforms. So that's the DNA platforms and the vectorized vaccine candidates. So first, starting off with the proven and widely used bucket, we have subunit vaccines. And so these contain pieces of the virus, and in this case, the spike protein, formulated together with an adjuvant to enhance the body's immune response to the protein antigen, because a protein by itself is not as immunogenic as the whole virus. But that's definitely supplemented by the adjuvant. And then when we look at the different spike protein vaccines in development, there are two types. First are truncated spike proteins. So those are shorter portions of the spike, usually containing the receptor binding domain. And then we also have full-length spike, which for a number of reasons we believe to be potentially advantageous. So when we look at the full-length spike programs in development, we can see that the majority of the programs are sponsored by smaller companies without proven track records or marketed vaccines, and also really not in partnership with large vaccine companies. However, in this bucket, Novavax's nanoparticle vaccine in particular is worth highlighting as being part of Operation Warp Speed and substantial external funding from CEPI and BARDA. And while they don't have a marketed vaccine yet, they've previously seen success with their platform in a large phase 3 trial for flu over other traditional flu vaccines. So we have reason to be optimistic about this approach. And then just today, as you can see, they're already in phase 1-2 clinical trials. They've released data, initial serology, immunogenicity data from these trials that look really promising. And then we'll get to that later after the walkthrough in a comparative basis with the other vaccine candidates in the lead that have recently released data. So Novavax is a candidate, full-length spike candidate worth following. Now moving down the map to the truncated spike candidates. As you can see here, the majority of these programs are also sponsored by smaller companies that will have a hard time, or it'll be challenging for them to scale up to meet global demand. However, within this group, Sanofi's truncated spike vaccine program is worth highlighting because Sanofi is one of the major international vaccine developers of the world. They generate a huge amount of flu vaccine every year. And while they're slower overall in the COVID race, they're still in preclinical development. They have guided to being at 1 billion doses by year-end 2021. And so because they have all the infrastructure in place necessary to really hit the ground running, we expect them to be part of the global solution, maybe not part of the first wave early next year, but shortly thereafter in 2021. So we just covered Sanofi and Novavax as promising subunit vaccines to follow. And just to round out our discussion of proven and widely used vaccine candidates, we get to the whole virus vaccines. And we're not going to spend too much time here, except to say that this platform is really a hedge against the possibility that neutralizing antibodies against other parts of the virus, so not just the spike protein, might be important for protection. And as you can see here, most of these candidates are sponsored by programs in China and India. And so at the moment, we don't know if that theory is true. We don't know if these vaccines work yet. But even if they did, the challenge surrounding this platform is really getting up to scale, to growing up enough live virus to generate enough vaccine to immunize the world. So largely these programs will be focused on their respective domestic markets 2021. So with that, we've gone over all of these vaccines to test the theory that mucosal immunity, or generating local IgAs, is important for generating sterilizing immunity and also herd protection. So we don't know if this theory is true yet, but we'll gain that insight from these programs as they pan out. So moving on to traditional injected adenovirus vaccines, we can look at the programs themselves in development. And as you can see here, some of the major players include J&J and their Ad26 vector, as well as AstraZeneca and the University of Oxford's ChAdOx vector. So the important challenge facing this platform is that people can develop immunity against the viral vector itself, which is why these major programs are using vectors that are less common. So people have had less exposure to these vectors versus CanSino or other programs using a more common version of the vector, Ad5. This means that we can expect J&J and AstraZeneca's program to be effective and part of the first wave of vaccine solutions. However, moving forward, this platform is not ideal for redosing or boosting. And just to get a general idea of where these programs stand in the COVID race, AstraZeneca is probably one of the leaders in timeline. They have already, so they're part of Operation Warp Speed, substantial external funding, and they have already pledged hundreds of millions of doses to be available between the US, EU, and India and China by the year end. So that wraps up our discussion of vectorized vaccine candidates. Now moving on to nucleic acid vaccines. So these include both DNA vaccines and mRNA vaccines, and we won't spend too much time today on DNA vaccines because mRNA vaccines are generally thought to be more immunogenic, and there are definitely more programs going after this approach. And when we look to these programs, we can see again that the majority of the groups in development are sponsored by smaller companies or earlier players that will have a challenge scaling up to meet global demand. However, when we look to the main players or companies with mRNA vaccine candidates in development, we can see here again that the major differentiating factor will be the final effective dose that these vaccine programs take into larger phase 3 trials. Because if you can imagine, if there's a limitation on how much vaccine that you can produce, and for instance, Moderna is going into their large phase 3 trial at 100 micrograms, if these other companies that are testing lower doses are effective, and then it will have a advantage in the terms of scale. So that's definitely worth following. And in particular for Moderna and BioNTech, their programs that are recently released data in this past month that we'll cover shortly. And then just to end the walkthrough of the structure of the map, the last piece to touch upon is the very bottom of the map, where we have all the more complex novel vaccine candidates. And we won't spend much time here except to say that there are a couple of programs backed by CEPI or even in collaboration with a large pharma vaccine company, Merck. However, we believe that these approaches, they have additional inherent risks. For instance, with this VSV vector, it's replication competent and poses risk to immunocompromised populations. And so as a whole, given that there are other options out there that don't have these inherent safety risks, we believe these to be at a disadvantage. And then, so with that, we've gone over the structure of the map, and hopefully you have a better understanding on our take on the advantages and disadvantages of the various platforms out there. And then the final question that the structure of a map teaches is, based off of these programs, when might we expect these vaccines to start coming online and how much will be available? So we track all of this in a milestones table, where we list the number of courses per quarter that these companies have publicly guided to providing, assuming that all of these vaccine candidates work. And from that, we can build a picture of there being hundreds of millions of doses available starting year end of this year, progressing to billions globally year end of 2021. And then on the second half of the milestones table, we also track the clinical timelines of these vaccines. So when they start trials and when we might know from efficacy data if these vaccines work. And so for now, the leaders are still just enrolling and starting off their phase 3 trials, but we're developing a snapshot from recent data readouts from phase 1 serology data, how these candidates stack up against one another. And I'd like to turn the mic over to Peter for a discussion on the data so far and how these vaccines look. I think you're still on mute, Peter.
See that. Okay, thank you. And Shanshan, if you could just review the safety data that we put together. I want to make sure that I know what was missing and what was actually really low, like on swelling and things like that. I was just reviewing it. Okay, so let me throw up a picture of my screen here. Okay. So we really had to scramble to fit in the Novavax data here and compare it to the other companies. And what we're showing you here are the neutralizing antibody titers for each of the vaccines as well as their control arms. Now, the control arms for these trials, what they did was they got some samples from people who had recovered from COVID. Right, and the assessment is, all right, if you recovered from COVID, then whatever antibodies you've got are probably indicative of the amount of antibodies you need in order to protect you, right? And so what you want to see is that a vaccine is generating at least as much neutralizing antibodies as an infection does that you are able to recover from, and ideally more, right? You want as much neutralizing antibody as possible. Now, the control arms for these trials, what you can see is that they vary. CanSino's is undetectable here. University of Oxford and AstraZeneca, their control arm generated neutralizing titers. I don't know what that is. That's like 50 or something like that, very small. And their vaccine generated less than that, right? So that doesn't seem very good. In the case of Pfizer and BioNTech, their control arm showed that their assays were detecting neutralizing antibody titers that looked to be, looks like about 100 or something like that, and their vaccine generated more than that. So that's promising. But it also depends which recovered patient blood samples you start with. If you take people who recovered from a very mild infection, they may have a very low neutralizing antibody titer. If you take people that recovered from a really serious infection that were hospitalized, they're going to have a much higher neutralizing antibody titer, right? And so we look at this, we see that Moderna, for example, had slightly higher control titers, and they did even better than that, right? So that's reassuring. And then what's interesting here is for Novavax, they had really high neutralizing titers for the samples that they analyzed from recovered patients. And the company indicated that they had quite a few hospitalized patients that really recovered from serious COVID. And so it may be that they're showing us some of the strongest neutralizing antibody titers that are out there among recovered patients. And so when they show that they're much higher than that, that's even more reassuring. So the relative difference of these two is actually comparable. Moderna is about 4 times higher than its control, and Novavax is about 4 times higher than its control. But if you had to choose what you'd rather be 4 times higher, I'd rather be 4 times higher what is already a high amount of neutralizing antibodies from what appear to be more seriously infected patients. Now, there can also be differences in the way that you run these neutralizing antibody titers, the actual assays. And so I think until you actually take these samples from the different companies and run them in the same laboratory on the same assays, it's hard to know exactly who's better than whom by how much, right? But directionally, this all indicates that these vaccines are better than better than recovering from an infection. the vaccines are basically giving you the kind of immunity you get from recovering from a really severe infection, and maybe even better than that. That's good. These are measured, really merely weeks after vaccination. In some cases, a week after your second dose, in some cases, 2 weeks after your second dose. And what you want to know is, what will your neutralizing antibodies be 6 months later or a year later? So it's best to start with as high a neutralizing antibody titer as you can get, given that you're going to want it to last for as long as possible. Now, it's also kind of interesting here. We tried to indicate with this black bar down here about what the neutralizing antibody titers were that Novavax got to after just one shot of their vaccine. And it looks to be, it registers. It looks to be about 100, which, it's hard to do cross-trial comparisons, but it certainly looks like it's better than the AstraZeneca vaccine, right? And so generating some neutralizing antibodies in patients after one shot is useful because, if you have to wait 3 or in some cases 4 weeks between vaccine doses, there may be people out there who go and sort of engage in some riskier behavior, stop social distancing, maybe go see people after their first shot, right? And so it would be good to know that they're protected or at least partially protected after that. And it looks like Novavax does generate some neutralizing antibodies after one shot. So that's reassuring. Ultimately, we'll see, from the protection studies, whether something like AstraZeneca, which is just a one-shot vaccine in and of itself, which is a good thing, right? People only need one shot, whether that ends up being protective. But if not, all these other vaccines do get you to higher neutralizing titers, albeit with 2 shots. So BioNTech gives you 2 shots, Moderna gives you 2 shots, Novavax gives you 2 shots. So now let's take a look at how tolerable these vaccines are. I'll skip over this. So in the case of the various vaccines head-to-head, well, can't quite compare them head-to-head, but we put the data on these same charts. We show you the first bar is Novavax, and the second bar is actually the placebo in Novavax's trials, because it's useful to realize that some things like pain or headaches, they occur, even when you don't give somebody a vaccine, right? And in the case of this sort of orangish color, that's moderate. That's a moderate headache. And you have a higher rate of mild and moderate headaches in just the placebo arm than you do of headaches after the first shot of Novavax's vaccine or Moderna's vaccine, right? Now, when you get to the second dose of the vaccine, so this is after the second Novavax shot, after the second Moderna shot, after the second BioNTech shot, you do get to higher levels of headache than in the placebo arm for Novavax, right? So what I see here is that like Novavax and Moderna don't really look like they're causing headaches in people after the first shot, but they do cause headaches after the second shot. Same deal here for fatigue. It looks like you've got pretty mild, fatigue levels for these vaccines, but after the second shot, you get to higher levels, particularly for the mRNA vaccines from Moderna and BioNTech. You get much higher levels of people experiencing fatigue. But notice here, after the second placebo shot, you can have a severe case of fatigue. That's pretty weird. Severe, that would be like grade 3 fatigue. Yet that's just, maybe a person's fear, that they might have gotten a vaccine. So as we look across here, what you see is that the first shot of mRNA, it hurts a lot. So does the second. You see fevers emerging from Moderna and BioNTech after the second dose, like systemic fevers. Even AstraZeneca's appears to cause fever, if this is correct. Shan, are we absolutely certain about the data in all these charts?
Yes. So you mean yes. So if there's nothing there, that means
that's right.
the
There's
rate
only
0.
one dose for AstraZeneca's. That's right. That's why it's there. So AstraZeneca has only one shot. Therefore, you don't look at their second dose. It doesn't apply. Though I would be curious why we have a second dose of AstraZeneca here. Did they have a 2-dose arm in their trial as well? They did, didn't they? That's right. They had a 2-dose arm. Okay. And then when it comes to swelling, we're not showing anything here for Novavax or for AstraZeneca. Is that the case, or are we just missing some data?
No, so those values are 0. The only thing that Novavax doesn't show is chills. Those are not applicable.
Chills. Okay, so in this case, Novavax didn't report chills. That's an adverse event that others measured, but I guess Novavax did not. That's actually reversed over here, where you see that Moderna and BioNTech did not measure tenderness, but Novavax did. So not all companies will look at exactly the same adverse events. You can look at something like joint pain, and you see after 1 dose, there's no signal here of joint pain, but after the 2nd dose, it's something that people can sort of think they have, even if they've gotten a placebo. But it does look like the vaccines have a higher rate of joint pain. I wonder how people sort of interpret joint pain versus muscle pain around the joint. It might be in the shoulder if that's where you got your shot. You have to really look at the marketed vaccines, and I wish we'd included that control here on the previous tour. We included Shingrix, which is the most difficult to tolerate vaccine out there that's commercially available, and it puts into context these kinds of adverse events are ones that are tolerated in a commercially available vaccine. I think it's reasonable to expect that if and when these companies start working on booster shots, they're probably going to work on lower doses of their vaccine, or at least the adjuvants that are in them, because it's easier to give a booster shot than it is to get somebody from 0 to 60, right? Like to get them vaccinated, from a standing start. And so I wouldn't be surprised if the seasonal recurrent vaccines that we end up getting down the road to boost our COVID immunity are better tolerated than this. Now, when you think about combining, a COVID vaccine with a flu vaccines actually are a combination of 4 vaccines. We vaccinate against 4 different strains of flu every year. And so you have to think about giving 5 times as much vaccine in order to have such a 4 flu plus 1 COVID pentavalent vaccine. So flu vaccines are called quadrivalent because they've got 4 antigens. And so when you combine in a flu antigens with COVID antigen, you'll have 5 antigens. And so we'll probably call that a pentavalent vaccine. And in the case of a protein-based vaccine like Novavax's or also Sanofi's, which Shanshan told you about, you can give more protein without giving more of the adjuvant. So you don't have to give more of this immune stimulating agent that is really responsible for a lot of the side effects that you see, a lot of the tolerability issues. But in the case of mRNA, and this is really interesting, mRNA is its own adjuvant. That means if you want to give 4 flu antigens, of mRNA, then you're also giving more adjuvant. And what you can see in the data that Moderna and BioNTech and Pfizer put out is that when they go to higher doses of their mRNA than what we're even showing you here, you get much worse tolerability. It's really hard to see how you could develop the mRNA vaccines to be pentavalent vaccines that cover flu. So I think that they're going to be hopefully good, really good for getting us out of the pandemic and giving us this initial protection. I struggle to see how they're going to help us with our annual, booster shots of flu and COVID. And in the case of the adenovirus vaccines, like AstraZeneca's or later J&J's, those generate immunity against the viral vector itself. So if you think of a viral vector as being kind of like a thumb drive that has the code for how to make the COVID vaccine on it, imagine if the more you use the thumb drive on your computer and you put it in, the more your computer basically rejected the thumb drive, it became immune to it and eventually would spit it out. So you could no longer deliver the code on via that vaccine if you kept giving it year after year. So that's the weakness of these adenoviral vector vaccines, that they're really not good for recurrent, annual booster shots. So this is what leads us to think that the future COVID plus flu market, which, if you consider that flu is about a $4.5 billion market, you give it about a 25% increase in price because you're adding one more antigen. And then imagine, two to three times as many people want to get vaccinated post-COVID. You're talking about 4.5 billion growing to being maybe a 10 to $20 billion market. When you think about that annual market for pentavalent vaccines, you're really looking at companies that are more traditional, like Sanofi and like Novavax, that have the wherewithal, the right platform for creating that. And Novavax actually got good flu phase 3 data just as the COVID pandemic was hitting. So they have that component that they can later combine with their COVID vaccine. All right, so I'll pause there and actually pass it on to my colleague, Jonathan, to take you on a tour of the drug side quickly, and then we'll hit diagnostics.
Great, thank you, Peter. So I'm going to be covering the right side of the map, which is the therapeutic landscape. And on this side of the map, there is a lot going on. If you scroll through all the programs, there's over 250 drugs listed. But even though there's so many programs here that we're tracking, only two have had strong enough data to support their use in the United States, remdesivir and dexamethasone. And for the rest of these drugs, the jury's still kind of out on whether they will provide benefit. So we're awaiting these data. So in this section, we have three main approaches: targeting the virus itself, and then in red, the other main approach is targeting the immune system. And I'll focus on this top one, targeting the virus itself. So in this bucket is where remdesivir lives. And remdesivir is a drug that targets the virus directly by blocking its ability to make copies of itself. And as a result, it is able to shorten the duration of disease symptoms. Remdesivir, though, is not a perfect tool. Its main limitation is that it must be given IV. And so its use is really limited to the hospitalized setting. But there's a whole other group of patients who have milder symptoms, and in which getting an intravenous infusion is not really amenable. So there's this second branch of drugs that are all related in mechanism to remdesivir, but are slightly more convenient to administer. And in fact, one of them is being developed by Gilead itself. It's an inhaled version of remdesivir that the company has announced and is now being tested in clinical trials. And then there are several other oral candidates that are also in clinical development. Just to name a few, favipiravir is being tested. Merck and Ridgeback are being tested as well in clinical trials, and Atea. And so all of these programs are being tested, and we should have some initial data in the next few months, discussing some initial efficacy of these compounds in treating the more mild form of COVID-19. And if we take a step back in the map, remdesivir is a type of drug that is blocking the replication, but there's also a set of drugs that are targeting viral entry itself. And the most notable part of this drug class is monoclonal antibodies. These are therapies that just take they're based on the naturally occurring antibodies from patients who recover, but manufacturing just the antibody. These are important therapies in development because they have the ability to both be used in the therapeutic and prophylactic setting. That is, so the use in the prophylactic setting can be important because it can be a stopgap before we have widespread vaccine availability It can also be used in patients who are potentially ineligible for vaccines due to autoimmunity or other reasons. Now, within this section, there's two main approaches that companies are going after. One is they're just developing a single antibody and testing it by itself, or other companies like Regeneron are developing a cocktail approach in which they're mixing two antibodies together. And we think that this kind of pooled cocktail approach is potentially interesting because it will allow an antibody, the therapy to potentially address any mutations that could emerge in the spike protein, and just potentially be more efficacious by combining. But just as an aside, an overall kind of view of the therapeutics landscape is all of these approaches need to be amenable to being tested alongside standard of care, because remdesivir and dexamethasone are already approved. Any additional future agents will need to be tested on top of them. So if an approach is not amenable to being combined in that way, it may not be able to be appropriately developed. With that, I will pass it to Ganesh to quickly cover the immune side.
Thank you, Jonathan. Let me share my screen. I believe, Jonathan, you have to unshare first before I can share. Thank you. So hi, everybody. I will be helping you to guide through the part of the right bottom side of the COVID-19 map. And we will be discussing the therapeutics which are developed for the critically ill patients. So 6% of all symptomatic patients can develop actually severe acute respiratory disorder, which can lead to respiratory failure. So it's great to have vaccines, it's great to have antivirals to target for moderate or severe patients, but we need additional therapies. On this end of the map, we actually look carefully on prevention of onset or reducing the impact of the cytokine storms. So the immune system attacks the virus and tries to cover it against it. But in the critical ill patients, we observe the signaling molecules get crazy and the immune system is overactivated. Therefore, there should be different approaches which would target this specific phenomenon. It could be either inhibit specific cytokines, or it could be targeting broadly the full inflammation. Here, we have observed recently in July by University of Oxford's clinical trial that a generic dexamethasone showed very effective results. It showed a reduction of mortality by 35% in ventilated patients, and further on 20% mortality reduction in the patients who would require oxygen. So this is promising and it's great. What are other approaches which could be helpful for us to fight and help the critically ill patients? The other approach would be targeting the cytokines, which lead actually the cytokine storm, which makes these patients end up with mortality. So specific cytokines were, a lot of people were giving high confidence with targeting the IL-6. So IL-6 has been a major cytokine which has been studied in multiple immune indications, and therefore we had already a couple of drugs which could have been tried immediately for COVID-19 as well. Unfortunately, Sanofi's Regeneron Kezara had already failed in July. And after our map update last week, Roche's actemra as well failed. This bodes, unfortunately, for the whole class that the targeting purely IL-6 signaling might not be effective for critically ill patients. So what does that leave us with? Recent clinical trial come out, which was targeting widely by immunomodulation of the overactivated immune system, which was the anti-6 antibody from Equilium Biocon. Biocon, the partner in India, had a small RCT trial with 30 patients where they could show 0% mortality in the single trial where they got approval for emergency use in India. For that therapy to be classified as completely effective, we will need a global trial where we will need at least more than 150 to 400 patients, which will have a similar result for potential US or EU approval. So we need several distinct mechanisms, as already Jonathan pointed out, which we could be combining with potentially generic dexamethasone or remdesivir to help the severely critically ill patients with COVID-19. Here we are tracking the 250 therapeutics drugs for the whole orange and red section, with including Jonathan's section, all the drugs programs where we are expecting data and when their trial started. So hopefully in this quarter and next quarter, we will get more positive data, which will enable us to fight with COVID-19 more efficiently with distinct weapons that could be combined with remdesivir or oral dexamethasone. With that, I would like to hand over to my colleague Parker, who will cover the diagnostic section of the map. Thank you.
Thank you, Ganesh. Okay. And you should be able to see my screen now. As of last week, we launched a diagnostics map to go alongside the therapy map that we just walked you through. And within here, we've got the roughly 150 programs that have all been granted emergency use authorization in the US and tried to put them into a meaningful context for you. So in diagnostics, we know there have been a number of issues as the field trying to keep up with the pandemic, but we start, as we do with all of our maps, in the center with a description of really how to think about navigating this. And really there are two primary questions that people face when it comes to COVID. The first is, who has COVID? Do I have COVID? And then the second is, who is already immune to COVID, with the surrogate for that being, who's developed antibodies to the virus. And the issues on these two sides of the map are very distinct. So on the question of who has COVID, the challenges there have really been about scale. And we'll talk more about really all of the issues that have arisen and the possible solutions that are on the horizon. On the right-hand side, it's been an issue of accuracy. So when you ask the question, who's already immune to COVID, you want to know that the answer to that question is being answered accurately. And on the left-hand side, I'll start to take you through some of the trade-offs that you should be willing to make in order to answer the question effectively. So with the question of who has COVID, here largely you're doing molecular testing, and everybody can follow in the news the ramp up that's happened in the scale of testing that's being done. But you also appreciate that it's not nearly enough. And you can see the rapid ramp here on the first chart as we've gotten to 800,000 or 900,000 tests per day. And just to put this in context, the tests that we're talking about here are molecular tests, largely being done by PCR or other amplification technologies. And if you look at 800,000 tests a day, why are we having trouble with issues like swabs and tubes and extraction reagents? And it really comes down to the fact that 800,000 tests a day for COVID alone now means that we're running more COVID tests than we are for all other molecular tests combined. So we're really putting a burden on the system that it wasn't built for. Additionally, a lot of those testing platforms are centralized in labs. And the reason for that is that tends to be the higher volume equipment. It tends to be the equipment that gives you the higher quality result. And in the past, when we've asked a question about a virus, when we've asked a question about, HPV or MRSA, it's been important to get that answer right. So we're willing to take a little bit of time and get it to the central lab equipment in order to get that answer right. And I'll talk you through some of the trade-offs that come into play when you're trying to control something like COVID, but getting it right exactly at the expense of time isn't always the best trade-off. So there have been a lot of plans around understanding what the scale of testing is that we need in order to open effectively, and we've consistently fallen short of those estimates. But I think more troublesome is that the vast majority of those approaches are really based upon either risk-based approaches or testing symptomatic patients. And when you have 40% of the population that has the disease walking around without symptoms, you really have to get outside of the mentality of a triage, and you have to get into the mentality of a screening program. So what we're proposing here is something that needs to be done on an entirely different scale, where you crush the spread of the disease while reopening. And that's really only enabled through routine testing on large portions of the population, basically anybody interacting with other people. And it makes you think a bit differently about the approaches that you're willing to entertain. And by thinking differently, what we're proposing here is this thought of a testing paradigm. And the premise here as you walk through this map is to think about how many infectious days am I pulling out of the system? And again, if you're not screening, then you're writing off the 40% of patients who are spreading the disease without having symptoms. So you really have to get into a screening paradigm. You have to get into that with some routine cadence. And what we propose here are ways to think about tools that we have in our arsenal that could deliver you a crude screen that would test the vast majority of people, and then reflexing to the high sensitivity, high specificity lab-based test. Or getting out there in the point of care with solutions that are fast, that can be deployed frequently, that can be run cheap and easy. And that really explains our involvement that we announced last week with the XPRIZE initiative and the COVID Apollo project, intended to accelerate options that really fill that void in the market. So this is our framework in thinking through trade-offs to get us to a screening paradigm that we believe is really required in order to control the spread. So given that scale is really the primary issue around answering this question of who has COVID, you'll see that the map on the left-hand side is really split, or the branches are really dictated by that scale question. So what we have here are all of the labs that are, coming and deploying all of the resources that we possibly have to try and answer this question within the traditional paradigm. But what's really needed are screening tests that change the paradigm. And that's where you see we've got these icons here for tests that would really get us into that screening paradigm that would help to crush while reopening. So that should help you in walking through the left side of the map. And I'll come over and quickly cover the right-hand side, which is the question of who is already immune. And on this side, really the burden is to get the answer right. And getting that answer right rather than getting it quickly is more important. And unfortunately, the first tests that came out, I think we're all well aware, were some of the point of care serology assays that give you an answer quickly, but maybe not correctly as to whether or not you have antibodies for the disease. So some of the early studies were done with these lateral flow tests, and they have 95% or 90% specificity. And 95% in a lot of cases sounds like a very good number, but when you deploy a test that has a 95% specificity into a scenario where very few people have antibodies to the disease, antibodies to COVID, you get a very high false positive rate. So here we illustrate that trade-off that was made in those early tests. And unfortunately, those early ones can put you in a scenario where if you only have 4% of the population that has COVID in a region where you're testing, and you have a test that's 95% specific, well, only 40% of those positive answers are right, and the other 60% are wrong. And that means that 60% of the people think that they've got antibodies. 60% of those positives think they have antibodies, think they're protected, and maybe looser with social distancing and other requirements to limit the spread. Whereas you really got to get out there with systems that are above 99%. And there you can be highly confident, even in low prevalence, that the answer is correct when you're saying a person has antibodies and therefore is likely protected. So in contrast to the left-hand side, you'll see the right-hand side is really completely centered around performance of those assays, and the performance being the most important parameter, and additionally giving you the ability to evolve those assays as more is understood about what defines protective immunity, evolving those assays by evolving the cut points. So you'll see the color coding as you navigate the map on that side will help you in understanding those trade-offs. And with that, I hand it back to Peter for close.
Sorry. Thank you very much, Parker. We packed a lot into 1 hour, and we're going to leave this active for maybe another 10 minutes or so. If people want to go through the Q&A section, we've answered a number of the questions that are in there, and we could try and tackle some more of them. And we'll keep accumulating the Q&A from these sessions and eventually put out a much more extensive Q&A, because we're seeing that some of these questions are repeats of what we've seen before. So stay tuned. We're learning from all your questions, and we'll keep answering these questions as they come. Thank you. I'll sign off with that.